Okay, sir, we are live now. You can start your session, sir.
Hello.
Yes, sir.
Shall I start off?
Yes, yes, yes. Yes, sir. You can start now, sir.
Shall I start off? Okay.
સર તમારે છે ને બે ડિવાઇસમાંથી અવાજ આવે છે એટલે એક ડિવાઇસ મ્યુટ રાખવું પડશે.
હા યસ હવે કદાચ નહીં આવે બરાબર છે
યસ યસ. ઓકે સર.
હવે આવે છે
ના સર
હજી
હવે
આવે છે
ઓકે.
અવાજ
ના સર હવે નથી આવતું.
ઓકે થેન્ક યુ સો મચ થેન્ક યુ okay I hope my screen is visible to everybody.
યસ સર.
Hello.
યસ સર. સ્ક્રીન ઇઝ વિઝિબલ સર.
Hello. Thank you so much. So shall we start now?
યસ સર.
Okay, so namaste friends. This is Dr. Sindhav Gauran, Assistant Professor at Gujarat University, University School of Sciences, Department of Zoology, Biomedical Technology, Human Genetics, Wildlife Biology and Conservation. Being a family member of the Gujarat's parents as well as an esteemed university, and yes, of course, the biggest university of the Gujarat, I would like to welcome you all to your coursework by the BKVNU University. Here, I would also like to convey wholehearted thanks to the team BKVNU for giving me the opportunity to connect, to interact, and share my views with you all wonderful friends today. Hope you all are safe and good in sound. As we all know, this time it's quite different and like a survival of the fetus, પરિવર્તન અને નવરચના જેમણે ગુજરાતીમાં કહે છે ને એક સિક્કાની બે બાજુ છે. So yes, of course, innovation and adaptation leads us to toward the learning. And yes, of course, the show must go on. We have faced many, ups and downs. We have seen the corona, and yes, of course, the many more things, the geopolitics and everything. But yes, the show must go on. So please, I request you all to join me to inaugurate my today's, interaction with a little warm up. So, this is, as we all know, sometime, somewhere, we all have, experience this question in our life, rather in any situation. And yes, of course, I must say, most of the time when we are weak or in some dilemma, the glass philosophy. Is the glass half empty or half full? And yes, we have been called depend on the our opinion, our options, and rather at the time maybe the glass half empty, sometimes we say glass half full. And is whatever we have been called, we have been classified, we have been grouped as an optimist or maybe as a pessimist, depend on our simple observation at that time. As many motivational speakers inspired us that we have to be optimist only. And yes, of course, why not? We rather we should. Why not? We must. Why? And but as an inner scientist, if we see it's a full of sciences, if we think beyond the line, it's always there. It's always something. How? Let's take a look. As a thinnest scientist, hope I'm making such things clear. We can see always glass full in the air part or in the water part. Yes, it is always. Just we should have a faith. We must have observations. We look into detail as we are practicing since as we start our life. And yes, of course, with the science, we all know. So yes, being a thinkers together, I'm confident we will have a great journey and enjoy ourselves together for next, up to 12. I'm giving the time up to the 1.5 hour. So together, let's start. So yes, something more interesting. I would like to, interact with you all, friends. So yes, here I my simple message. Please have a look at this, this short clip, and having this, short clip. I mean to, I request, try to think, try to speculate. Let be the thinkest. અત્યારે નહીં તો ક્યારે નહીં અને કલ કરે वो आज करे वो अभी कर. So what this clip is about, you can ping in chat box, or you can take down your opinion, right? And what we have look into here, I hope each and every individual, right, each of us have many opinions. Here I must say, we all might have different perception, different opinion, different point of view. Today we not gonna classify like many of us observe beauty, wonder, plant, yes, of course, the nature, and yes, of course, as a science, the growth, and yes, whenever we don't have any answer, it's up to God and whatever it's all about, God's creation, etc., etc., and many more. You all are correct in one way, or I must say in another way. For what we are doing this, and most ultimate and one common question is, what is life? And together, life is the aspects of growth, reproduction, activity, change, process, acts, reactions, adaptation, survival, mortality, and of course, many more and many more. Together, let's see events as a life. Today, what I'm interacting with you just as a novice, my small journey with you, and it's all around the life. And yes, of course, we all know the importance of the life, and as a ultimate Homo sapiens, we work for theirs, directly or indirectly. And yes, of course, in my life also, I also used to have the same questions. And yes, not only the one question, many questions, many doubt. And as for what is life? However, to surprise, I really found the answer from my favorite Scarlett Johansson, not directly, as I'm a diehard fan of her. And what the movie Lucy, many times. I hope you all have gone through the movie Lucy. Very scientific, very fundamental, movie. And just to have her, I watched many times. And literally, I don't remember how many times I have seen that, movie, for just to have her only. And I'm watching her, but she answered. She answered about the life, and a life lesson. As you might see the presentation in the movie by a great actor and the role played by the scientist, the Morgan Freeman, in the movie about the life. It means a lot. How? Let's see. So I understood two terms over there, and here, as you can see on the screen, the first is a immortal, means living forever. And it mostly not possible by anyone in any situation. Really not possible to date. In future, we may, of course we may. And we all are, I can say, no one doesn't know about the future. And as I mentioned, in future it may be. However, we all have an idea. The cancer cells classify in this category. And yes, as we work with the cancer cell line, it's still today it's a growing HeLa cell line. We all know about this, and even using many the fundamentals and many theories. But yeah, till date we must say it never possible to have a lifelong. And yes, of course, we have the aging in parallel, and the life ultimately have the death also. So the next option we have is reproduction. So what is it? The action or process of copying something. The production of offspring by sexual or asexual processes is mostly performed in a favorable environment only. And this is the only basis of diversity or evolution in life. And the ultimate option for any species is to be immortal, as my great great grandfather, my great grandfather, my grandfather, and my father is in me, and also me now in my child too. As humans, the most selfish species on this beautiful earth, this is what we consume. We have developed a very favorable environment, and by adding in or implementing the second phenomena, we are now more than 9 billion in probably a few years. And an event of reproduction lead us to transfer of this hereditary material to the offspring from parent. We all know by what we all are very well know, the DNA only. Here by this magical paradox, I must represent this very fundamental, as we many a time see the Homo sapiens sapiens is the most powerful and the most intelligent species on earth. But yes, by this slide, I'm very confident you're going to respect the nature, as we all have studied. A single cellular organism, a yeast, you can see in a favorable environment when having the fundamental nutrition, when having the favorable condition, this species goes for the mitotic division. And yes, whenever the atmosphere, whenever the environment is not quite favorable, it choose a meiosis. And yes, of course, it's always a survival of the fetus. And you can see how smart it is, how fundamentally it live on earth, right? And it do understand by single cell only. So this is the life. And today my the small journey, we would like to soon understand what I mean to say, what do you mean by life. And yes, the life is ultimately what. And I'm very sure today we're going to see the life as some different opinion, by understanding a different options and what should be rather importance. So yes, let's see further this. I hope by this example I'm going to clear my chalk domain by domain. So yes, as I mentioned, this all fundamental things ultimately work by the paradox, a fundamental, a magical molecule, the DNA only. And yes, the DNA is a magical paradox entity in the whole universe, what I understand till date. Sometimes when a child has achieved the best, we used to say, Mara jeo che, mere jaisa hai, like me only. And likewise, many more things can be a similar between the parents and child. That is called inheritance. Passing of trait like physics to the disorder. It can be our height, color, nature of the disease. This is about the similarity. But some differences are also remarkable. And what we have today, contrary to inheritance, it's a variation. And variation is a unique in a brood. We all know we are having some distinct feature, sometimes very powerful, sometimes maybe some compromise, but ultimately that our parents don't have. This is variations, and this variations is results of reordering this magical paradox, that is a DNA. And if it is spelled correct, we are so lucky. Please mark my word here. We are so lucky if we have this sequence in correct in way. Even it's possible to get some good supremacy also, as you can see on some speculated sci-fi movie like X-Men, Marvel, Avengers, and many sci-fi movie. But unfortunately, what happened if it is misspelled? One has to suffer. One has to pay its consequences. And today I'm going to give you my small journey on such phenomenon only. And the term mutations suits both conditions. However, this misprinted mutations can accelerate some lethal genetic disorders. In crux, here my ultimate message is the science of genetics, the science of heredity. The science of heredity or genetics is the study of two contradictory aspects of a nature and a nurture. And yes, I here must admit the great oncologist and author of the book, The Gene by the Siddharth Mukherjee. He mentioned very fundamentally by this phenomena between nature and the nurture. So yes, it's play an important role in performance of the new species. And yes, I must say the science of hereditary genetics is a study of two contradictory aspects. The hereditary, as you can see, the inheritance, what we are term as similarities, and on the other side we have a mutations that leads to the variation, and ultimately what we are together now and what we nowadays have a say the supremacy of the Homo sapiens sapiens. Here I must say why and don't know why and how we are understanding all these things. Just I recently said a single unicellular organism, a yeast, do have all this understanding, but I think uh, they may never utilize the earth like this or having such the things. But ultimately, let's see. So yes, today my a small story, the story of DNA, the molecule of the life, is a journey, and we all are very well aware about this molecule, right? And as I mentioned, the central dogma is a journey of amazing discovery. It began when scientists like Theodore Soman and the Antony van Leeuwenhoek used their microscope to see the tiny cells and invisible life in a water. Later, Frederick isolated a mysterious substance from the nuclei of a white blood cells, calling it nuclein, and what we now know as DNA. Though its role was unclear then, this was a pivotal step in unveiling the molecule that carries the blueprint of a life, whatever today we are. The real breakthrough come one day. A woman named Rosalind Franklin captured a crystal photo that held a big secret. Using her image, Watson and Crick figured out DNAs, a twisty ladder shape, unlocking how life passes on its code. And yes, we all know about this code, the central dogma from DNA to RNA and RNA to ultimately protein, and what this blueprint. Ultimately, a surprise, the parts of this code were found in meteors, suggesting life, a building block, may have come from a space also. But yes, of course, it ultimately, in some, the DNA holds the power to shape a life, and possibly it control too. Used wisely, it can cure disease too, but in wrong hand, it power could change destiny. DNA is just not only a molecule, it's a key to life and to our future. So pertaining to above, I have compromised this talk on this, DNA disharmony, a genetic disorder, what I'm going to mention you all. So yes, I have uh, a few things to share with you, and of course this is the topic where I'm working since last decade. And પેલું ગુજરાતીમાં આપણે કહે છે ને કવિ એની કવિતા વગરના કે ને સાયન્સ ટેસ્ટ એના સાયન્સ ને કીધા વગરના આવે. So on the same ground, just kidding. I really very this topic is really very very near to my heart, and its journey is started since last decade, and to work with this condition really little known and lethal too. This condition name is an Duchenne muscular dystrophy and abbreviated as a DMD. What is all about? We'll definitely see soon. And we all have an idea about the central dogma, as just I have mentioned. So once this DNA, the godfather, order our little chefs, the RNA, start to prepare food as a protein and for our body and the functionality. What we till now, I hope I'm clearing, and we all know this is in very well. But what if an order get jumbled? As I mentioned, the variation, the mutations. You can see in this slide that functionally will no longer exist like before. and leave us with a question: why me? We can set a term genetic disorder here. And yes, if I say in India, more than 50 million individuals are affected by various genetic diseases, and this number is going to mount up. Uh, Many times, as genetic predisposition in common diseases is more common toward our Indian community, in this genomic era, the population has restored their health and avoided unnecessary therapeutic by with the help of proper, and as mentioned, prevention is better than cure. So you at least have some things in our hand. And at present in India, one of the biggest gain is the openness or availability of such molecular diagnostics and molecular tools and techniques. Gujarat also has a fundamental role in this, and therefore in the information which to develop a strategy, especially the DMD, BMD, and we'll talk with you all wonderful friends today. So yes, as an introductory, I understand the two term over here. One is an U. We mostly understand this U and NU in our routine, reading time. So U is all about the evenness, and NU is something without evenness. So we ultimately say when such things are uneven. So in genomic, in our blueprint, when such even and uneven may be have, it leads to the some genetic condition, what we ultimately called about. So that's maybe eugenics and the NU genetics. And why I'm emphasizing this phenomenon, we'll see later on. And this is very important to understand. And yes, as I mentioned, the topic and my journey I'm going to share with you is now I'm going to put front of you all friends, my journey with the muscular dystrophy, where I'm interested. And what is muscular dystrophy? So muscular dystrophy is a broad group of hereditary, progressive, degenerative disorder of muscle, which affect both humans as well as the animals. The word dystrophy is derived from a Greek word, which means difficult or faulty, and the trophy, which ultimately we can say a nourish. So muscle weakness is a primary symptom of all this of the muscular dystrophy together, and clinical and pathological criteria have been used in the past for classification, but nowadays we are using such a fundamental and the very high throughput tools, the molecular tools and techniques that ultimately help us to understand the root cause. So on basis of that, this muscular dystrophy now have been in this, I must say, in this genetic era, these are the what we have this rare condition, like the variation over the 40 genes to 34 different type of muscular dystrophy nowadays, depend on their clinical features, inheritance patterns, severity, and yes, of course, the affected target protein. The main culprit here is a DGC complex, that is a dystrophin glycoprotein complex, and their various segmental protein. So any of the protein, if it do have some mutation, it ultimately leads to the condition we term as a muscular dystrophy, which is crucial for fiber stability. In this clinical entity and the broad term, today I'm going to discuss with you a very frequently found here, I must say, and yes, of course, the most lethal, most severe condition among this all muscular dystrophy, that is a Duchenne muscular dystrophy. And this Duchenne muscular dystrophy also have it less severe allelic form, that is a Baker muscular dystrophy. And this together we term as dystrophinopathies. So this is what I'm going to mention you today. So what is DMD, BMD? As I mentioned, it is an X-linked recessive disease, which ultimately affect the 1 in 3,500 live male birth. Affected individual can have mildly delayed motor symptoms, as you can see on a slide. These are the symptomatic portrait of the patients. Will later on definitely give you the more point on this part. And yes, as per my experience, I still understand the clinical symptoms at the forefront. if you understand the clinical symptoms. And yes, that's why in my initial sites I have mentioned the pessimist, optimist. Yes, of course, everything is fine. I'm never mind for this. But yes, as observations, what realistically we are seeing as a thinkers, what we are observing, that ultimately helps us. And yes, a scientist named Duchenne ultimately identified this condition 2 decades ago by simple observations. So as you can here also see the calf hypertrophy, you can have on look, you can see the Gower sign, what it is all about. I'm sure sharing with you all friends. As I mentioned, this is a X-linked condition, ultimately affect the male child. And the gene, if we are talking about, it ultimately located on this, the wheat gene. Of course, whenever we are talking about any genetic diseases, we must know the location. And the gene ultimately, so which gene or genes are responsible for this? So the answer is the longest human gene, that is a DMD or a dystrophin gene. The metagene of the human responsible for this DMD and a less allelic, that is a BMD, Baker muscular dystrophy. So ultimately as a dystrophinopathies, because the responsible gene is I must say here dystrophin gene. The condition was ultimately first isolated gene by the positional coding method in 1986. And the official name of this gene is as I mentioned, the dystrophin gene, and the symbol is a DMD gene, as you can see on a screen. And its location is, as I mentioned, the X chromosome. And in the X chromosome, we categorize to understand the location of the gene. So that's why it's XP 21.2. P and Q are the arm of our chromosome. So for our sake of understanding, we have categorized. So the upper is a P and the lower is a Q. And as you can see, this is the ultimately what we can have an idea about this location. And by understanding this location, as you can very well see the things here. You can see this is a P arm and this is a Q arm. And ultimately in this P and Q arm, in the P arm at the band 21.2 is the sub band 2 and a band 1, and the region what we are talking about 2. So the chromosome location is ultimately XP 21.2, what we have used to say. And yes, this DMD, as I mentioned, largest known human gene, is highly complex, covering 2.4 million base pair with 79 exons containing at least 8 independent tissue-specific promoter it do have. And this ultimately, if you are talking about the consisting of this almost 0.8% of the total human genome and about 1.5% of the entire X chromosome. This is the big gene. It is the DMD gene. So yes, this largest known DMD gene provide an instruction for making a protein called as a dystrophin protein, and it ultimately it do have a various tissue specific isoform also. So as I mentioned, you can very well see over here, these are the our muscle tissue. And if we look into in detail, you can see this is our sarcolemma, and how this red color dystrophin gene ultimately work as a shock absorbing, or you can say the anchor protein to our muscle stability, and it provide ultimately the muscle to the strength. And this together, the whole, it term as a DGC complex, as I mentioned, dystrophin glycoprotein complex. And as you can see, apart from a dystrophin, we do have many other protein like dyspherylline, psychoglycan, laminin, collagen. And yes, if we found mutation in such the particular gene also, this ultimately leads to the condition, the other type of muscular dystrophy, as I mentioned, maybe the limb girdle muscular dystrophy, maybe congenital muscular dystrophy. So yes, this ultimately the DMD protein found in a muscle fiber membrane. It's a helical nature, allow it's act like a spring or a shock absorber. Dystrophin protein is located primarily in a muscle used for movement, that is a skeletal muscle, smooth muscle, and cardiac muscle. Here, this is very complex protein. So for simplify, I just make it a graphical presentation, so we can at least understand there the important segments like the N terminal, C terminal, and yes, of course, the very important in to understand this disease condition is the central road domain. Yeah, everything is very important, but we found most of the mutation in a central road domain. So yes, this protein is primarily in a muscle used for movement, and as I mentioned, it is in every muscle, skeletal, smooth, and a cardiac. And small amount of dystrophin are also present in nerve cells in the brain tissue. This protein, if we are talking about, is measuring about 150 nanometer. Is a rod-shaped cytoplasmic protein and is a vital part of a dystrophin glycoprotein complex. As you can see over how it anchoring the cellular segment with their sarcolemma segment, and of course with the support to directly or indirectly to the extracellular part also. So this connect the ultimately the surrounding, I must say in simple term, to the extracellular matrix with the cytoskeleton. So this dystrophin protein is primarily transcript of this, I must say, the transcript of this into the mature mRNA that is approximately about 40 kilobase pair, and encode a protein that is a 420 kilodalton approx. And by the amino acid participation is a approx 3600 amino acid residues will be there. This sarcolemmal protein comprises again the highest 0.02% of a total muscle protein, and yes, up to 5% of this membrane skeleton. So yes, in sum, this protein work as an anchoring and shock absorbing protein in muscle cell. Will make you more clear soon by having a small animation also with me. So yes, by understanding the basic and root cause, so ultimately our target is to have an answer. As I mentioned, the questions are always there, but yeah, if you have a cancer, the question, we can ultimately identify the various strategies, the mode by which, as I mentioned, the Homo sapiens sapiens always have the answers for anything. So here by again the same thing, the mutations and the queries. So what pathway we do have. So yes, we do have some diagnostic strategy. So as I mentioned, the clinical features are always at the top. As I mentioned, the clinical features are really the very fundamental things for us. So by having this, looking and getting this, the observing these clinical symptoms like calf hypertrophy, Gower sign, frequent fall, toe walk, we can definitely help us to diagnosis. Yes, the biochemical parameter, again a very important and fundamental thing, where by having this, the biochemistry, the marker molecule here, the total CPK, creatine phosphokinase can give us an idea to screen this condition before having this very expensive or hi-fi tools. The muscle biopsy is again a very fundamental tool, but yes, nowadays having this the molecular test, it is quite yes, it's still very important, but yeah, the molecular test can give you the broader picture. As of course in diagnosis, everybody think of having the non-invasive test, and yes, the electrodiagnostics are always at the top. So sonography and MRI can give you the idea, and same way the imaging test will which also help you out. And yes, this is the molecular test where I'm much interested, and I'm going to give you the idea about this in a broader way and in a more in deeper way we are going to discuss about. So yes, this is what I'm trying to mention you about. This is the Duchenne muscular dystrophy. It's one of the under the umbrella of the muscular dystrophy. Yeah, this is the one kind of a Duchenne muscular dystrophy. And yes, to understand, and yes, of course, I must say we are coming to the most interesting part of this interaction, and I'm sure you will also appreciate or like it. So what it mean by, so it gives you a glimpse of the condition, and we of time don't even look at them early. So let's begin by just watching a very brief animation movie. What we have, try to pile up in a 2-minute animation. You show a whole series of activities, symptoms, life of this Duchenne patients that might you observe somewhere. I mean, when one child start to fall down, what a mental condition might the parents have to face when they know how their now their child age is not more than 30 years. He will be wheelchair bound by the age of 12 to 13 years. We forgot that this is just not one child story, it's all about one family. It's all about the entire societal structure might face. I actually want to show you how much of tough for the family to take care of once a child. We do by labeling now more no more hope for because till date no such drugs or the therapeutics are available for such condition. So you might question then why one should with all such things. Here I must put that nowadays with molecular tools and the genetic techniques, if we can diagnose a patients early, we can have a child age quite more as well as less severity for later life too. And yes, in this genetic era we are having a few fundamental drugs for a particular type of the mutation in the market, and yes, few are FDA approved too. So we are hoping the same for this, Duchenne muscular dystrophy also. And we are dedicated to working in this field. And think everyone's brain is now swinging. And let me play this short clip for this condition to understand. And together we have made for this for the mass awareness and educational purpose. Hope you'll enjoy this clip. Let me start. Just a second.
A young gentleman and a beautiful lady were eternally bound by the holy ties as Saptapadi Mitra. In the coming years, they were given the loved gift of parenthood and were blessed with a baby boy who was the center of the universe. And the sticking laughter filled up their lives with unparalleled joy. Time flew by as the growing boy excelled in all areas of his life, and the family was happy with his achievements. One day, during a cricket match, the mother noticed that her boy started having difficulties keeping up with his friends. He seemed to walk much slower than his peers. This made his mother tense and worried about his son. Soon the boy's parents noticed that their child had weak calf muscles, had difficulty climbing the stairs, and had frequent falls while walking. The tears of joy were soon replaced by uncertainty and tension. Then came a day when the young boy had to use his hands and arms as a support to stand, and eventually lost his entire ability to move.
Now very interesting part is coming. Yes, this is we tried to give some positive message. So this is all about the our story of this Duchenne muscular dystrophy. And hope you enjoy it and have now you got a scenario or this what is about the DMD, how we can identify it. So it's like a simple boy story. It start by simple, I must say, the awkward movement, ultimately the wheelchair bound, right? And yes, this is the what we observe it in initial level. We can have a early diagnosis. This is the what awareness brochure we have prepared for the and what ultimately the how we manage this condition, it's all about. So yes, muscular dystrophy, as I mentioned, is a group of inherited diseases that damage and weaken a muscle over a time. This damage and weakness are due to the lack of protein, as now we all know, the dystrophin protein, which is necessary for normal muscle function. As you now can see with the small animation what I have mentioned. So it's like simply by to have one the boat to anchor in a big ocean. We used to have one a simple anchor to have, and this anchor can give you the stability. But yes, this anchor ultimately bind by a one very strong rope in a chain, right? And if ultimately it get broken down, this ship may not get stability, and it may be anywhere in the ocean. So this is the same condition what we have found with this, dystrophin protein. It's work like a anchoring protein, right? And yes, which is necessary for normal muscle functioning. And this absence of this protein can cause a problem, as now I hope you all with aware about. Yes, this is about the core or actual problem with the DMD, and it's as I mentioned, the largest human gene providing instruction dystrophin protein, and that ultimately together strengthen the muscle fiber, protect them from an injury, and working as a anchoring protein. And ultimately, it's provide the muscle the strength. So yes, what we work out. So our study is started by the journey to understand this the patients. And yes, as I mentioned, currently there is no such fulfill therapy or drug exists, so except for some palliative type. And yes, recently we have a few drugs which are in conditional trial, and by hoping this all those things we would like to work. And during my research I came to know some finding. As you can see in the slides, majority of patients show the deletion in the downstream region, and the frequency of an individual exon deletion is presented. Exon 50, as you can see, which was the the highest frequently found mutation in this patient. And if you are talking about the deletion number 45 and 45 to 52 are the two big largest gene deletion we found in our Gujarat population. And this is ultimately, as I mentioned, the protein part. It's in the central part of this the protein. And the pattern of mutation found in Gujarat is as you can see in the Gujarat map. And yes, in our study cohort, out of many familial cases we also have observed. And yes, by having, and why such so things, yes, we have also established one a PCR technique with a term as a multiplex PCR assay. So the diagnosis quite in cost cutting also, and in some speed up way. So in some, the study was carried out in a limited number of these Gujarati BMD patients, and the method was MPCR is doubtless a good tool, but it not ultimately identified every of the mutation. Thus, we ought to use an alternative method like the MLPA, that is a multiplex ligation dependent probe amplification, or yes, of course, a direct sequencing, the NGS, next generation sequencing, for diagnosis such a small, or you can say the point mutation. And yes, if having a carrier recognition in early stages, we can definitely have many things in our hand. So we are also get some carrier recognition. The location and size of a deletion mutation found in this dystrophin gene do not indicate any correlation with the severity of this DMD and BMD condition, but we ultimately understand the very basic phenomenon of our genetics if we found a frame shift mutation. So you can say the out frame mutations, and ultimately leads to the Duchenne muscular dystrophy. And if it is like an in frame mutation, we can predict kind of a BMD, that is a less severe Baker muscular dystrophy. But yes, I here must admit, we do have a few exceptional cases. And yes, having the outframe mutation, they are living like a BMD condition. So such the exception, which is helping us to find out some good therapeutic molecules. One of them is a utrophin molecule. So we are hypothesizing, and yes, in the country, in the world, many people are behind this hypothesis of the utrophin upregulation, that at least convert this DMD condition into the BMD condition. As I mentioned, the biochemical indices, of course, do not have a potential role in diagnosis, even though they can be a capable enough screening and management of the patient. And this study strongly emphasized a need for further investigation into genotype-phenotype aspects of this Gujarati DMD-BMD population in order to provide a better diagnostic, prognostic, prenatal services, and counseling to the suffering family. So yes, as I hope now you have a same query, why me and why somebody, and yes, by having this much of hi-fi and this much of time, what ultimately we get. So yes, in this genomic era, we have a vast data of human genome after this successful completion of this Human Genome Project. And this Human Genome Project gives us the ability for the first time to read nature's complete genetic blueprint. And yes, till date, we are having a many fundamentals that we are doing. And yes, ultimately, this blueprint is all about how human was made and evolved. And yes, by having the fundamental databases like the OMIM, that is the Online Mendelian Inheritance in a Male, that is abbreviated as OMIM, is a comprehensive, authoritative compendium of a human genes and genetic phenotypes that is freely available and updated frequently. The full text reference overviews in OMIM contain information on all known Mendelian disorders. Duchenne muscular dystrophy is also one of them. And over 15,000 genes, such variations and such the mutations database are available on this website. And OMIM focuses on the relationship between phenotype, genotype. In sum, we can say it's a guide, it's a mentor in understanding the genetic diseases. So now, as I mentioned you earlier about the DNA, a magical paradox, I must say, monomer in the genetics. So for understanding genetics, or rather say a life, so let's see the DNA. So DNA is present inside, as we all know, every cell of our body, which do have a nucleus. Many cells does not have. It's fine, because their ultimate goal is not to be the survival part or the reproduction, as I mentioned. So each cell contains roughly 3 meters of the DNA. If we unravel all our DNA from all of the cell and laid out the DNA end to end, the strand would stretch from the Earth to the Moon many times. The DNA is as we all know, this much of big data, this much of big, the DNA. How can be in a single small nuclei? So yes, this is the DNA, and the DNA is tightly coiled up and structured into a 46 chromosome that we all know about. And our chromosomes are arranged in a pair that we have, as you can see, these are the chromosome, what we have in our in our karyotyping. We used to say, when we are talking about the human, what we have a 46 XY for a male, and if the female we check, then the 46 XX. So here you can see X and Y, and this 23rd pair. So ultimately what we say, the Homo sapiens human have the 23 pair of a chromosome, and the total number is a 46 number of a chromosome. And yes, this last pair we term as an sex chromosome. And this apart from this 23 pair, the 22 are termed as an autosomes. And they all do, as you can see, have their respective gene location, as we have discussed with this this X chromosome. And I hope now you all know this is the what we know about this. This is the X chromosome. And this Duchenne muscular dystrophy, if you're talking about, this is a P-arm, and this is above is what we term as an this is as an Q arm. And if you're talking about the gene location, is quite over here, right? XP21.2. So this way this is very helpful to us. And yes, if you are talking about the similarity, so of course we get the one chromosome from the father and the another from the mother, and ultimately we are the diploid. So these are the chromosomes which are always in pair. And yes, this ultimately give you the, as I mentioned, the genetics, the study of the two contradictory aspects. One is in variations and another was in inheritance. So that's why the inheritance, we get the half from a mother and we get the half from our paternal side. So this is ultimately what we are looking about. And yes, as I mentioned, the inheritance pattern. The inheritance pattern is very important in the, in our, to understand this, any of this genetic conditions. So here, as you can see, in this genetic condition, as I mentioned, this is a X-linked recessive condition. So this is X chromosome in a parents, and the Y chromosome of that male parents, and the female containing a XX. That's why the 46, XX. And yes, of course, the heck is O here. If you see the female, it possesses two X chromosome. So having one chromosome with such mutation, with such alteration, can fulfill the part of that mutation by the another chromosome. So that's why we used to term as a mother, the female as a carrier. So see tentatively a non-symptomatic. But yes, if you see the inheritance pattern, if we see the ratio, with this couple, we can definitely have a normal boy, normal female. But yeah, again, there is a chance of 25% having a female as a carrier. And yes, a 25% chance of having a boy, a male boy having a Duchenne muscular dystrophy. And of course, the male part don't have the supplement as an extra X chromosome. So there is the lack of this protein, and ultimately we term this condition as an Duchenne muscular dystrophy. But having an understanding of this, carrier mechanism, which help us to maintain this condition, and can be very well understood by getting the idea of these, familial inheritance pattern. So this is the how this basic and fundamental things help us. So as I mentioned, each cell contains roughly 3 meter of a DNA, and we have one, as I mentioned, as a diploid. So one chromosome from a father and the one from the father, so half. And there are approximately, if you are talking about this later, these are the very important later, that is now we term as an A, T, C, G. In the DNA, in the code is made up of this 4 letter. As our computer system work by a 0 and 1 binary language, our body, the life, is worked by this 4 letter, that is A, T, and CG. And when we're talking about the human genome, it contains 3 billion such letter, 3 billion such chemical later, and that are very massive. And this is a massive amount of information. It would fill approx 200 of this, the yellow pages in a small type font. And if you try typing the whole genetic code, it would take years. And yes, it would be without any breaks we have to. And that's why nowadays we have such hi-fi next generation DNA sequencing and everything. You may heard about when we are giving the data for a DNA sequencing, they ultimately ask for the TBs of the data, how much GBs of the data we are having from such hi-fi DNA sequencing machine. So yes, this DNA is made up of these 4 building block that we now know as a ATCG, and help us to grow, develop, work, function. And what we are today, ultimately, I am very sure now you all understand, just because of this 4 later. So once again, altered the fault in our gene, and then it is tough life, nothing is important now. And that ultimately what we are talking about. And now might you have a question on how we can study such genetic disorder. I tell you, it is nowadays quite very simple. To let me share my a small experience with you all, wonderful friends, as it involves a step by stage, the SOP. And first and very important domain is a proforma. To have a proforma feeling, the proforma is what? The proforma is ultimately individual specific, disease specific, some information together, some forms which we can identify some our observations. And yes, This proforma can help you to narrow down the condition. As I mentioned, if you have a Duchenne muscular dystrophy kind of a condition, by having their familial history, by having their previous report, you may get some CPK value in their previous reports, and you can add such particular specific biomarker in your proforma. So this will help you out to get the idea of this Duchenne muscular dystrophy. And same way on another kind of like in a I must say in a cystic fibrosis, right? So it's a condition with the lungs. So by having their marker understanding, the proforma is very particular and very specific, and which help you in very fundamentally. You can also note down your observations during your first station. The patients can have a Gore sign or not. What about the toe walk? What about the age of onset? Right? Such things having in a proforma, which help you in a lot way. And yes, as it is always say, the very important domain in your proforma, it's a pedigree understanding. What do you mean by a pedigree? So pedigree analysis is very important tool. And yes, as an Ahmedabadi, as a Gujarati, it's quite a free, and that's why I and rather just for the kidding, but it is really important too. And if you know the proforma and the pedigree in a very deep down way, the half war can be definitely win by this two domain only, right? So the pedigree can give you the idea about the familial inheritance and all about the familial condition. So before starting anything else, it is very important to have the empathy as well as the faith and the confidence. Proforma will help us to get a detailed information of the subject, as I mentioned. And moving to the pedigree, it is very basic and fundamental. And as I mentioned, it's a pocket-friendly, practice to trace any genetic condition in a family, in a race, in a population. And we at our home even have a practice this, and yes, we all know the AMBO system. Whenever we have a marriages in the family, we used to check the gotra and labla thing, and how we have this AMBO, the familial tree, what we have. This is ultimately the scientific pedigree analysis, the scientific phenomenon of to have the family tree, and this family tree can give you the lot of informations. So yes, as I mentioned, our parents used to see a family tree for marriages, and why such practices? This is a very fundamental practice of the eugenics. We'll soon have a look on that also. So yes, they were so right, and this is the concept of eugenics. As I mentioned, what do you mean by eugenics? We'll see, and that's why I'm emphasizing in my initial sites about the word "you." Here you can understand eugenics, euthanics, euphanics, and how we'll talk and what we can pass out the message to the family. That is very important. So for further analysis, the sample collection is very important. Rather, it must to have. And as I mentioned in genetics, and of course, the two fundamental the approach you can have, the non-invasive and the invasive. It's always preferred to have a non-invasive. Yes, as I mentioned, the DNA is everywhere in a body. So by the buccal swab, it may work. By the blood, it is also works well. But yeah, the buccal swab is quite a non-invasive, so it's a quite preferable practice. But yeah, sometimes somehow we have to take a tissue-specific sample. So non-invasive we have to also perform. And yes, as I mentioned, the radio immuno, the imaging test can also helpful to screen out the condition. But yeah, ultimately, to identify the root cause, to understand such genetic diseases, we do have a two broad domain. One is in the cytogenetic domain, and another one is in a molecular domain. In cytogenetic domain, we have the karyotyping, we have a FISH analysis, we have a MFISH analysis, where you are focusing for the chromosomal part. If any big mutations, deletion, duplication, inversion, ring chromosomes, such things on a broader level, we can definitely work out with the karyotyping system, where we are quite focusing to understand the chromosome first. And yes, when you have a small mutation, so we can say at the gene level, the mutation at the ATCG, the kind of code, that where you need some high-fi molecular tools like a PCR machine, that is a polymerase chain reaction, invented by the great scientist, the Kerry Mullis. And we all know the power of the PCR machine. And after having the PCR machine only, what we are today and what we are nowadays understanding in this genomic era, all credit goes to the Kerry Mullis. And that's why this PCR, the polymerase chain reaction, is also famous as a people's choice reaction. The scientific journal is very well termed it is as a people's choice reaction. So yes, and PCR is a very basic and fundamental tool. If you are performing a next generation sequencing, again it do require a basic PCR to having. Yes, but nowadays this Oxford and PacBio system, they quite not require the PCR machine, but ultimately to have such things, the PCR is quite till date it's very important. And at my lab also, we are working this PCR by the modification of MPCR. The MPCR again is what? The MPCR is again ultimately the multiplex PCR. So within a single PCR reaction, we can simultaneously identify the multiple targets. So that's why the multiplex PCR is very famous, as is pocket friendly, cost effective, and quite the time saving. So this is what we understand the as a SOP, like we can now very well say. First is you require a standardized prescribed proforma, and yes, of course, in these conditions, the ethical approval are also very well important, and you have to have work with the ethical institutional ethical guideline only. So this proforma needs to be approved. Your proforma must have such marker symptoms. The pedigree analysis, the definitely very helpful. And yes, ultimately the sample collection by the phlebotomist or by the hospital and the clinicians. And we always work in a good collaboration with the clinicians only. I work with the our great helping hand pediatric doctor, Dr. Anil Sasar. He helped me a lot in every of my research step. And he is really quite a good mentor to me also in this, to understand the clinical condition. And yes, by having all those, the parameter and the SOP to follow. And after diagnosis, still there, of course, we used to say a patient, yes, of course, this is a diagnosis. Now the mutation is there. And ultimately the patients are there. So they ultimately have no hope for better health or like the life ahead. And yes, the again question same, the arise after all such so hardworking, after so much, money, spending, and as I mentioned, what is life and why me? So again, the patients will definitely ask you, the why me only in this world, and what is life and how to manage it. So that's we used to have the approach. That's and yes. Here there comes a very crucial element in a genetic diagnosis, and it is termed as a genetic counseling. We do have a good genetic counselor also with us. So genetic counseling is a responsible communication process with a consultant, or rather say a proband. Here you may have sometime heard this word, a consultant and a proband. So consultant by mean what? So whenever somebody found some such condition in their family, and they seek to have for their life, and to have some suggestions, some idea for their life, or for their the offspring, or for their family, the individual we used to term as a consultant. But the proband is the subject, the individual who is suffering with this condition, right? They term as a proband in this this whole the practicing. So as I mentioned, in genetic counseling, you may have to face with the consultant or a proband, right? So in simply we can say, if you have a Duchenne patient with in the process, the Duchenne muscular dystrophy patients, if we consult, then we term him as a proband. And yes, their mother, father, we used to term as a consultant, right? And the proband's brother, the SIPS, if he or she asks for their, for the life ahead, we term him as a consultant. So as I mentioned, the genetic counseling is a very important and a crucial element, is a process through which knowledge about this, genetic aspects of the illness is shared by the trained professional with those who are at an increased risk or either having a heritable disorder or passing it on to their unborn offspring. A genetic counselor addresses and concern of the patients, their family, their healthcare providers, and support a patient and their family dealing with this, illness. So as we can see in this slide also, the proper responsible under the ethical with the all of the privacy, the discussion is the good options. And yes, ultimately in genetic counseling we say the word empathy is very important. So you have to be with the patients in very empathetic way. You have to work in a empathetic way. And yes, as I mentioned, the privacy is quite very important in this such condition, right? Because it's a familial matter. It's suppose the thing of directly with the patients. So this is very, and yes, always it is a suggestive in manner, not at all in an advising manner. It's a suggestive. And ultimately our target is to make them potential enough to have them the understanding of each and every concept of their condition, and to make them aware about, to give them a knowledge about this condition. That is a main role of this genetic condition and counseling. And of course it's play a very big role, to maintain such condition, to have to take care, to manage such dreadful conditions. And yes, the very very fundamental options are, as you can see, I have put so many picture of many known personalities on these slides, because they accomplish many things in their life and with challenging times, and become epitome, or rather a role model for many of us. Here are as a team to have strength for such a, so such things can be helpful as empathy. We all know the case of this Angelina Jolie, right? As her family is diagnosed, or they have some high risk of this breast cancer. So such a great and fundamental personality goes with the mastectomy. We all know Washing Akhtar, great bowler, but yeah, having the diabetes, but having a good, the counseling, the managing part, he or she can done a lot, right? We know the John F. Kennedy, the asthma he do have, but yeah, of course he work on a very fundamental way. We all have studied the royal family, right? How hemophilia can be a dreadful condition. We all know about the Amitabh Bachchan, the thalassemia minor, and lucky having the Jaya Bachchan. But of course, nowadays the techniques are there. If they're having the thalassemia minor partner, having few test availability, you can have a quite good role. So yes, by having such empathic approach, we can strengthen them. We can definitely give some hope. And as a human, the different from the all species, we are curious and problem solving. Here the ultimate aim to pass the message that is never give up. And because no one can beat the person who won't give up, so that's where it can work. It is very well work. And such patients are also living a very fundamental life ahead. And yes, here what we have started, I'm quite near to winding up. So yes, since last few years, working as an educator, as a novice, small researcher, having a goal for a best life for all of us, and especially the Duchenne muscular dystrophy, I do believe these three approaches together, and by heart, because as it is and most fundamental approaches for the better mankind. As in my initial slide, I also have given you the idea as you and NU. So I hope now we are very well understand you. It's mean the evenness, the harmony what we have. So if your chromosome, all chromosome are quite more in number or quite in less in number, so we used to term as an U, right? Euploidy, right? So that is the all about how many times of they may increase or decrease. Aneuploidy, it's all about the unevenness. From our 23 chromosome, maybe any one or two, or maybe number get increase or decrease. Like we can see the trisomy. So trisomy, it's all about the 21 we all are aware about. The trisomy 21, a Down syndrome. So we used to term as an aneuploidy, because in whole set of this 23 chromosome, only 21 number get in increase in number, right? So that's why we term as an aneuploidy, so unevenness. Euploidy, we all know the wheat, it do have a hexaploid, the whole set of chromosome now 16 times, right? So this is the evenness and the aneuploidy. So ultimately these words are quite very fundamental, and in these three terminology what I'm talking you about, these three terminology are euthanics, euphenics, and the eugenics. And yes, what I understood by my small research and understanding, and by reading such things, I found the everything in close proximity with this three terminologies. So first, let's understand the what do you mean by euthanics. So yes, as you can see, the euthanics, what when we're talking about the euthanics, which is the study of the improvement of the human functionality and well-being by improvement of the environment, better nutrition, education, and yes, with sufficient amount of the medication. So this is the approach what we used to term as an euthanics. We always try to have such a good environment, less pollution. So these are our efforts is wholesome as a term as an euthanics. We are always strive for a better nutrition, better fundamental of the education, and yes, when we are having some minor issues during our life, what we have a good and yes, a less toxic, a very fundamental drug to utilize, right? That is the phenomenon is termed as an euthanics. I hope now I'm clearing what is life, and in the life, the euthanics is again a very fundamental approach to have the improvement in our life. Yes, this is a very fundamental approach, eugenics. What do you mean by eugenics? So eugenics is improve the future generation by improving the germplasm of this existing population. In this both hereditary and a suitable healthy environment, it is a very important and play and significant role, as I mentioned to you earlier. As any species have such condition, they try to be immortal. As not possible directly, so one another option, that is a reproduction. And as we discussed in prior, and hope you all are now agreeing with me, play an important role in developing a eugenics, the social claiming to improve the genetic feature of a human population through selective breeding, encouraging the marriages between the desirable candidates, what we term as positive eugenics, right? So this is all about the eugenics based on the idea that is possible to have distinguish between the superior and the inferior element of the society. Nowadays people are trying to, but yeah, of course, what we are trying for, like the selective desirable, in animal husbandry we also have seen, right? So this is a very very fundamental of this eugenics option. The third and very fundamental options, then the approach where I'm focusing, it's a euphenics. So what do you mean by a euphenics? So yes, where we have to do a lot, right? I must say this is indeed required nowadays. So yes, the symptomatic treatment of a condition, and here I must say a symptomatic treatment of a genetic diseases of the any flora and the fauna. You all know the gene therapy. We all have understand the bone marrow transplant. We all know the CRISPR-Cas. We know the embryo therapy. We know the molecular medicine. Such phenomena are nowadays, I think, we can put into the euphenics. And yes, we know the some issues, some error are always there, and euphenics is definitely help us out to resolve such mutation, such error. So yes, I'm confident that you also now believe, and at least once please think with these three concepts for a better world. Let give the best world to the next generation. In sum, here I must add, and also you all have marked that all these three fundamental a prefix like the u, and it's mean the even. So it is definitely make the human life even and more fruitful ahead, the having this as a through together, right? So with the above ground, we have a plan or better say to the hypothesis on which we are working. And we are still struggling with the many things, but yeah, we are working for a betterment of this muscular dystrophies, especially the very very lethal, and as you can imagine, these patients. And yes, this patient, when we are conversing with them, when we are discussing, because nowadays they all are our family, right? So when we are discussing with this patients is ultimately mentioning you, now I have only 5 years to live. You see the conditions, the dreadfulness. And yes, the family, the patients have to suffer a lot, and this is all about the DMD condition. And it's not in only our Gujarat, it's everywhere in the world, right? And there is no any bar, no any poor, any rich, any hi-fi, any geographical. It's everywhere in the life. And as I mentioned in initial, it's also in a human as well as in the animal also. So by putting with our full energy and blessings from the Almighty and all you wonderful friends, we are doing quite a few things. And yes, this is what we researching right now with as an wholesome. So as you can see, our main target are ultimately the to find out the patient first from this big population. So as you know, we have some brochures with us. We have some this 2-minute animation movies available in a Google. You simply YouTube search a Duchenne muscular dystrophy by Gujarat. You can definitely get our research article and this YouTube link also. So this is a simple awareness by having the idea of especially the Gover sign. The Gover sign is a clinical sign to identify this Duchenne muscular dystrophy, as you have shown all in the video also. So this is ultimate sign is what whenever the patient wish to stand up, he should rather must put his hand to his knee. This marker, the uplifting, is ultimately termed as an Gover sign. And yes, by having such screening of the patient, we do the biochemical analysis. As you can see, as I mentioned, this what we have developed it at my lab. This is a very fundamental things. What we have, work out. This is a multiplexing, the MPCR. And this such things is help us to cost cutting as well as to simultaneously detect the many more disease condition. And this is a tissue histography we have to sometime have to do. And this is the all about the chromosome what we understand. So ultimately our focus is to understand the genome part and to get some proteome part also. Here at this juncture, I'm happy to share we do have a one project from the ANRF, where we from the Gujarat University, myself, and I'm very lucky to have the mentor like Dr. Sharad Gupta sir from the IIT Gandhinagar, who helped me with all his efforts for the proteome part. Nowadays we are focusing on the proteome part also for such especially the hotspot mutation of this Gujarat. We do also focusing on some biochemistry, as I mentioned, the total CPK. We also try to get some non-invasive parameter from the urine ultimately. And yes, we have also adapt our Indian legacy as a Prakruti, as a Ayurvedic approach. And yes, literally, I'm very happy to share. We found a very ultimate and very fundamental results with this, Duchenne muscular dystrophy, that our article is also published in a good Ayurvedic journal. If you search a Prakruti analysis and a Duchenne muscular dystrophy, you will definitely get from the my scholar profile as well as in the Google. And where we really, I must admit, by having such Prakruti understanding, we are real time managing these patients, right? So that is a good things what we have right now. And ultimately, as I mentioned, we are trying to have some U part in this this in NU part of this muscular dystrophy. Some unevenness mutation is there, so we try to manage them. So we are right now with working with this hypothesis. And ultimately, yes, here I must say in whole world, this 7th September, it is celebrated as a Duchenne Muscular Dystrophy Day. And this whole September month is a famous as a muscular dystrophy month. We also do many activities for the awareness, for the upliftment of the patient on every basis, on the health basis, on societal basis. We do, as I mentioned, this is our small brochures where we can, circulate to the society. So which help them to identify, as I mentioned, this is a clinical sign of a Gower sign. You can understand. Now we can sum up like a calf hypertrophy, the frequent fall, the breathing difficulty, the toe walking, waddling gait, and ultimately the cardiac and the respiratory movement leads to their, the death. Yeah, we also have seen yoga and the physiotherapy. These are the good options for their, the management part. And I hope now you all know about the diagnostic approach. We do have some observationals, clinical, biochemical, and molecular diagnosis are very well help us out to understand this, condition well. So for the welfare of this, DMD patients, we are working all those phenomena. And yes, we have, we will work with the Indian Muscular Dystrophy Society. We have more than, thousands, probands as a family with us. We try to, as I mentioned, elevate the condition of our, patients by providing them supplements like L-carnitine. We have seen very good effect of the nutraceuticals molecule, that is L-carnitine, to such patients. They can avail a physiotherapy, wheelchair assistance, BiPAP machine at our, the society. We help them for a special needs also. Whenever we are able to, we are regularly, organizing such, vaccination camps, awareness programs, and the, cultural functions for their, shake of encouragement to their, social interaction. So these are the few glimpses of the our efforts, what we have worked out, and at the broader way, as a small my research journey. And yes, I'm happy to share recently our Gujarat data, what we worked since last the decade. It is now available on the Leiden database. Leiden database is also one of the, database library, where you can identify such, all over world, whatever the mutations are available in the world. So this is, of course, we are now having such, data. I'm also thankful for the scientist Dune Duchen, who help us in having this, database incorporated into these, Leiden database. I'm very thankful to the scientist Leiden. And yes, at the last, before say goodbye, aujo, sayonara, the take home message for all of us. And yes, I know my starting journey question, what is life, is yet not answered. But before that, let's have some take home message for all of us. So please take care of your, both the very important entity in the life. So that's your DNA. And for the second, I hope you all get my point. And it is really important, as you can see, both the cases. Once it is out of, control, once we did any mistake, then there is no second chance. And I must say, even God cannot help us. So I must say, take care of your DNA as well as the better half. And so please take care. Here I would like to acknowledge my university, Gujarat University, my Department of Zoology. I work with the IIT Gandhinagar in collaboration, Indian Muscular Dystrophy Society, the funding, NRF, and of course, today for this platform, the BKVNMU University and the whole team of the Life Science Department. I'm very much thankful for especially Dr. Professor Suhas sir, to give me such a nice platform to interact with you all wonderful friends. And of course, I must acknowledge you all of wonderful friends. I hope we'll see you soon in somewhere, in somehow, in some. You all are welcome to my lab also at our Gujarat University. I'm thanking you all for all of your time, your support, and of course to listening me, to give me the chance to share my small research journey with you all wonderful friends. These are the few references where we work. And yes, ultimately, in a conclusion, from when we started our lecture with the question, what is life in a nutshell? I think you all now agree with me. The life is a 4-letter word. Of course, LOVE is always at the first, and life again a four-letter word, LIFE. So LIFE, LOVE. But yes, of course, I hope today I make you the things clear. This A, T, C, G. This four-letter again do have a lot of importance. If a single letter get misspelled, if a single letter gets some jumble, the life may be very, very tough. And hope I justify today's talk, a title, because after having and doing all the above, we used to have the questions from most of us. Hope will soon have a good news for such tuitions, for such genetic mutations. And yeah, then of course I'm very sure in this also the Gujarat, India will be the harbinger to give the answer for all those conditions. Hope you all enjoyed. Wishing you a joy, happy, and a beautiful life ahead. Thank you so much, everybody. And so this is all about my research journey. Again, thank you, Professor Dr. Suhas sir, BKVNU University, and the whole team of this Life Science Department for giving me such an opportunity, this chance to share my journey with you all, wonderful friends. Thank you so much. Stay safe, stay blessed. Thank you all. Thank you so much.
Thank you, sir, for this session.
Thank you so much.
Welcome, sir.